Look at a sleeping dog lying next to a rock. To the naked eye, neither is running, hunting, or doing anything we typically associate with being alive. So how do we actually know the dog is a living creature? The answer requires us to abandon the idea that life is defined by the physical actions we can see. If we magnify our view of the dog, we bypass the illusion of stillness. Here, on a microscopic scale, molecules are in constant, violent motion. Living organisms are built from these molecules, assembled into highly ordered, complex chemical structures. But the surrounding environment exerts constant physical and chemical pressure on these structures. Without active intervention, this precise organization will inevitably degrade and collapse. To survive, the organism has to fight back. It performs a continuous, automated series of repair jobs, what we call life processes, to rebuild those degrading structures in real time. Running these constant repairs requires immense amounts of energy. The organism is locked in a perpetual cycle. It must continuously pull energy from the outside world and convert it into internal maintenance to stave off collapse. Because life on Earth is constructed from carbon-based molecules, the raw fuel an organism pulls from its environment must also be built from carbon. Plants and some bacteria bypass the need to hunt for this fuel. These autotrophs build their own complex carbon compounds from scratch, using simple inorganic materials like water and carbon dioxide. They drive this manufacturing process by absorbing external light energy from the sun. To gather the necessary carbon dioxide, specialized guard cells on the leaf surface act as mechanical valves. By filling with water, they swell and bow outward, opening a tiny pore to draw the gas inside. Once the autotroph synthesizes its carbohydrates, it stores the excess as starch, creating a stable internal energy reserve for future repairs. Animals and fungi take a different route. These heterotrophs lack the biological machinery to synthesize their own food. They survive by consuming the complex carbon structures already assembled by autotrophs. But a plant's carbohydrate molecule is too large for an animal's cells to use directly. The animal must deploy biological catalysts called enzymes to dismantle these complex structures back into simpler, absorbable pieces. Whether an organism is weaving molecules together using sunlight or tearing them apart with enzymes, the biological imperative is identical: secure the raw carbon materials required to keep the system running. A single-celled organism can absorb these materials directly across its outer membrane. Its entire body is in contact with the environment. But as body size increases and internal design becomes complex, that direct environmental contact is lost. The millions of interior cells in a human body cannot rely on simple surface diffusion to get their fuel. The solution to this scaling problem is a highly specialized internal transport tube, the human alimentary canal. The process begins in the mouth, where mechanical crushing is paired with salivary amylase to immediately begin degrading complex starches into simple sugars. This processed mass is pushed down the esophagus and into the stomach. Here, the material is subjected to an extreme chemical environment. Gastric glands in the stomach wall flood the chamber with hydrochloric acid, activating a protein-digesting enzyme called pepsin. Pepsin can only function in a highly acidic medium, making the hydrochloric acid a strict requirement for breaking down dense protein structures. To prevent this acid from digesting the stomach itself, the organ constantly secretes a thick layer of mucus to protect its inner lining. As the acidic mixture exits the stomach and enters the small intestine, it must be quickly neutralized. A new suite of enzymes is waiting, and they require an alkaline environment to operate. Liver bile floods the intestine to achieve this alkaline shift, while simultaneously emulsifying large fat globules into smaller droplets to expose more surface area. With the environment prepped, the pancreas injects a cocktail of enzymes, including trypsin and lipase, ensuring the remaining proteins and fats are completely digested. The inside of the small intestine is lined with millions of microscopic finger-like projections called villi. This is the final extraction point. The dense clustering of villi massively increases the surface area of the intestinal wall, allowing the now simplified nutrient molecules to be absorbed directly into a network of blood vessels. This structural design guarantees that the raw fuel is transferred into the bloodstream for immediate delivery to every cell in the body. This massive, complex digestive organ system doesn't actually produce a single drop of life-sustaining energy. It exists solely to extract raw fuel, like glucose, and prepare it for the cells to burn. Once that glucose arrives inside the individual cell, the energy is still locked away within its carbon bonds. A precise chemical reaction is required to release it. That extraction process is cellular respiration, the oxidation of food sources to generate usable energy. This chart shows the universal first step of the process. Regardless of the organism, the cell takes the 6-carbon glucose molecule and splits it into 2 3-carbon molecules called pyruvate. This initial breakdown happens in the cytoplasm. What happens next depends on the available environment. In the absence of oxygen, a process used by organisms like yeast, the pyruvate undergoes fermentation, converting into ethanol and carbon dioxide. But highly active multicellular organisms require a more efficient method. They rely on the aerobic pathway, sending the pyruvate into specialized organelles called mitochondria. Using oxygen sourced from the outside environment, the mitochondria completely dismantle the pyruvate. This leaves only water and carbon dioxide as waste products, while releasing a massive yield of cellular energy. Respiration is the ultimate chemical bottleneck. An organism can ingest and digest all the food it wants, but without this oxidative process, that fuel remains locked and useless. But simply releasing energy inside a cell isn't enough. If that energy disperses freely, it's wasted. It has to be captured and targeted exactly where the organism needs maintenance. This is where ATP, or adenosine triphosphate, comes in. It serves as the universal energy currency for the cell. The energy released from respiration is used to take an ADP molecule and force an inorganic phosphate onto it, forging a new, high-energy bond. You can think of this newly formed ATP molecule as a fully charged, rechargeable battery. It can now be transported anywhere in the cell. When a specific cellular mechanism requires power, the cell cleaves off that terminal phosphate linkage. This single break releases exactly 30.5 kilojoules per mole of energy, deployed precisely where it's needed. This highly controlled discharge is the driving force behind the critical endothermic reactions of the body. It powers muscle fiber contraction, the synthesis of new proteins, and the conduction of nervous impulses. The environment never stops tearing down the structural integrity of biological cells. The threat of decay is constant. Life is defined by the ability to continuously push back against that decay. Even when an animal is completely motionless and fast asleep, billions of cells are acquiring carbon, extracting glucose, burning oxygen, and spending ATP in a highly ordered chemical symphony just to keep the